Good day, science majors. This is Sheryl Ordoña reporting about meiosis. Meiosis, the form of cell division by which gametes with half the number of chromosomes are produced. The distinction between diploids and haploids is the number of chromosome sets found in the nucleus. Haploid cells have only a single set of chromosomes while diploid cells have two sets of chromosomes. Meiosis happens in sexual reproduction and it has two divisions which are the meiosis I and the meiosis II. Fertilization is the fusion of a sperm and egg to form zygote, which is the fertile egg. The egg, which contains 23 chromosomes, and the sperm, which contains 23 chromosomes also, combine and have a total of 46 chromosomes. Sex cells is divided to produce gametes or sperm and the egg. Gametes have half the number of the chromosomes. Occurs only on the gonads, testes, or ovaries. This picture shows how pair of chromosomes are distributed during the process of meiosis. This picture is taken from a mouse testes. We all know that meiosis occurs in sex cells, and this is a microscopic view of how meiosis occurs from one parent cell all the way to its four daughter cells. In interphase 1, similar to meiosis interphase, chromosomes replicate. Each duplicate chromosomes consist of two identical sister chromatids attached at their centromer. Centriole pairs also replicates. Before entering meiosis I, a cell must first go through interphase. This is the same interphase that occurs before mitosis. Let's go to meiosis I, cell division that reduces the chromosomes number by 1/2. It has four phases: prophase I, metaphase I, anaphase I, and telophase I. Prophase I is the longest and most complex. In here, chromosomes condense. Synapsis occurs. The homologous chromosomes come together to form a tetrad. Tetrad is two chromosomes or four chromatids, the sister and non-sister chromatids. Synapsis takes place during prophase I of meiosis. When homologous chromosomes synapse, their ends are first attached to the nuclear envelope. Then, the intervening regions of the chromosomes are brought together and may be connected by a protein RNA complex called the synaptoamenal complex. Homologous chromosomes, pair of chromosomes, maternal and paternal, that are similar in shape and size. Homologous pairs, tetrads, carry genes controlling the same inherited traits. Each locus position of a gene is in the same position as homologous. Humans have 23 pairs of homologous chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. Next is karyotype. Karyotyping is a method of organizing the chromosomes of a cell in a relation to number, size, and type. Homologous chromosomes. When recombination occurs during meiosis, the cell's homologous chromosomes line up extremely close to one another. Then, the DNA strand within each chromosome breaks in the exact same location, leaving two free ends. Each end then crosses over into the other chromosomes and forms a connection called chiasma. Humans have 23 sets of homologous chromosomes. Each homologous set is made up of two homologous. Autosomes are code for most of the offspring's traits. In humans, the autosomes are sets 1 to 20. The sex chromosomes code for the sex of the offspring. If the offspring has two X chromosomes, it will be a female. If the offspring has one X chromosomes and one Y chromosomes, it will be a male. In humans, the sex chromosomes are the 23rd set. The Y chromosome contains a male determining genes, the SRY gene that causes testes to form in the embryo and results in the development of external and internal male genitalia. The crossing over. Crossing over may occur between non-sister chromatids as the chiasmata. Crossing over segments of non-sister chromatids break and reattach to the other chromatids. Chiasmata are the sites of crossing over. Paired chromosomes from each parent align so that similar DNA sequences from the paired chromosomes cross over one another. Another way of meiosis makes a lot of different sex cells crossing over. Crossing over multiplies the already huge number of different gametes types produced by independent assortment. This showing the sex chromosome, XX chromosomes for female and XY chromosomes for male. During prophase 1, the complex of DNA and protein known as chromatin condenses to form chromosomes. A large structure called the meiotic spindles also forms from long protein called microtubes on each side or pole of the cell. Between prophase 1 and metaphase 1, the pairs of homologous chromosomes form tetrads. Metaphase is the shortest phase. Tetrads align in the metaphase plate. Independent assortment occurs. First is the orientation of homologous pair to poles is random. Second is variation. And the third is the formula 2 raised to n. As the example, 2n is equals to 4, n is equals to 2. Thus, 2 squared is equals to 4 combinations. Metaphase 1 is a stage in the cell cycle where all the genetic material is condensing into chromosomes. These chromosomes then become visible. During this stage, the nucleus disappears and the chromosomes appear in the cytoplasm of the cell. Anaphase 1, homologous chromosomes separate and move towards the poles. Sister chromatids remain attached at their centromeres. Anaphase 1 involves separating the chromosomes from each sister chromatids to the opposite poles still attached to the microtubes of the cell. Then telophase 1, each pole now has the haploid set of chromosomes. Cytokinesis occurs and 2 haploid daughter cells are formed. During telophase 1, the chromosomes are enclosed in nuclei. The cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into 2 daughter cells. Each daughter cell is haploid and has only one set of chromosomes, or half of the total number of chromosomes of the original cell. Meiosis 2, no interphase 2, or very short, no more DNA replication. Remember that meiosis 2 is similar to mitosis. Moving on to prophase 2, same as the prophase in mitosis. In prophase 2, the nuclear envelope breaks down and the spindle apparatus forms. The chromosomes becomes arranged on the metaphase plate much as the chromosomes do in mitosis and are attached to the now fully formed spindle. Then metaphase 2 is same as metaphase in mitosis. During metaphase 2, the centromeres of the paired chromatids align along the equatorial plate in both cells. Then in anaphase 2, the chromosomes separate at the centromeres. The spindle fibers pull the separated chromosomes towards each pole of the cell. However, in anaphase 2, same as the anaphase in mitosis, sister chromatids separate. Telophase 2, same as the telophase in mitosis, where nuclei form. Cytokinesis occurs, and remember 4 haploid daughter cells produced. Gametes is equals to sperm or egg. The cell pinch in the center and divide again. The final outcome is 4 cells, each with half of the generic materials found in the original. In the case of males, each cell becomes a sperm. In the case of female, 1 cell becomes an egg and the other 3 becomes polar bodies which are not used. And this shows how meiosis gives rise to 4 unique daughter cells, each of which has half the number of chromosomes as the parent cell. And that's how the process of meiosis takes place. Thank you for listening.